Most gracious God, thank you for allowing us to be together in your presence today. We are grateful that we can still come together in this manner, although we yearn to see each other in person and anxiously look forward to the time when we can. Oh God, you know that we are people of thinking and of action, but especially now we find ourselves powerless over circumstances around us, circumstances which are sometimes confusing, sometimes frightening, sometimes maddening. We know you are with us as we move through this historic time of change from what was to what is yet to come. Please remove the hurdles of overthinking and of too much busyness, which can get in the way of our listening to you with our hearts, and put us in a place where we are able to see simple solutions, which we could not have seen with our own intelligence alone. Then we can respond with confidence and compassion in the world and with our brothers and sisters in order to live out our motto, service above self. Amen.
Amen.
Thank you, Catherine. And David, can you bring up the flag for us, please? Thank you very much. And if you will, each of you join me in the Pledge of Allegiance. Pledge allegiance to the flag
of the United States of America,
to the republic
to the republic which stands
one nation
indivisible, with justice for all.
Thank you. On a little bit of a sad note, I need to report to you that Mark Sinclair has announced his resignation from the club. We're going to miss Mark. He's been a big part of this club for a long number of years, but he cited the inability to meet in person as a primary reason. And I hope that at some point, maybe when we can meet again in person, we can recapture him, if it will. But in any event, we do want to thank him for his contribution over the years and wish him the best of luck. In the meantime, as I have said earlier, we're having a joint meeting and we welcome the Thursday Club members. And Tara, if you would like to address us at this point, we would appreciate that.
Thank you, President Lou. I appreciate your giving me a little bit of time. You know, we're excited to have these shared meetings. We figured that we can really bring in some great content and great speakers when we do some things together. So I think we're going to try this 2 meetings every month, and your club is going to meet with our club on the 21st of January, and we look forward to that meeting. We're having a presentation from Alex Lee that day about their expansion. So that'll be great information to hear about good things happening in our community. And the thing that I really wanted to share with you is the exciting news of our 100th anniversary, our centennial celebration, which will be held on February the 11th. And that will be a joint meeting with your club as well as other clubs and a district-wide event. We're excited about some dignitaries that are coming from the district level to speak with us that day in celebration of our 100th anniversary. We're going to have some proclamations and different things happening, as well as a historical slideshow, and just exciting time to share with each other that day. Of course, it will be virtual. And so with that being said, though, with a virtual program, you can bring people from farther away. So we have our fingers crossed that hopefully Rotary International President Holger Nyack will be able to join us that day. So that will be an exceptional program for all of us to share in. And District Governor David will be there with us, as well as a few other speakers. So just excited about that. We'll send out a separate link to everyone for that time that day. And then hopefully late summer, early fall, we will have some type of event to celebrate that centennial celebration. So we want everybody to be there and to participate with us. That's all I have. Thank you for the time.
Well, thank you for that, Tara. And that sounds awesome. You've got some real good plans in the future here. So we'll certainly be looking forward to that. Before we get into our program, are there any other announcements that any of the members need to make at this time? Okay, hearing none, we'll get right to the program. Today, our program is on Pfizer's COVID-19 search, I think, for the vaccine. It will be by Dr. John Earl, and he will be presented to us by our fellow Rotarian, Jeff Mackey. Jeff, you have the floor.
Thank you, Lou. Our program today is presented by Dr. John Earl. Dr. Earl did his undergraduate studies and attended medical school at the University of Oklahoma. He did his residency at the University of Virginia. He moved to Hickory in 1976 and founded Hickory Family Practice. His partners in that practice included Jim Hodges, our own Lee Young, and the late Will Thompson. He retired from that practice in 2016, and in 2020 became the primary investigator for the development of the Pfizer vaccine in Hickory through PMG Research of Hickory. John and his wife Jane have 6 children and at last count 17 grandchildren.
Wow.
In his spare time, he serves as a deacon at St. Albans Episcopal Church. John is an avid reader and occasionally shows flashes of brilliance on the golf course. He and Jane also enjoy traveling. Lake Hickory members may remember from my program last year that they joined my wife Florence and me in hiking 48 miles in 4 days along Hadrian's Wall in England in 2018. A piece of trivia about John: his father sang at the wedding of Roy Rogers and Dale Evans. I give you my friend, John Earl.
Thank you, Jeff. Let me get my screen up here and we'll get going. Hope everybody can see that. I just want to thank you all for inviting me, and thank you, Jeff, for inviting me to this meeting. This is a very important topic, and it has impacted all of us in certain ways. And what I'm going to do today is just basically a little bit of overview of some background material with COVID. What clinical trials are and how I got involved, and some of the experiences we've had with the trial and how they were done.
John, your presentation isn't up yet if it should be.
It's what?
It's not up yet if it should be.
Oh, it says my screen sharing is paused. Okay.
Yeah.
Oh.
You want me to just cancel the screen share and have you start again? Would that be quicker, John?
Yeah, why don't you do that?
Okay. Now, why don't you try again?
Okay.
There you go.
Yeah.
All right.
Just start your slideshow and you should be good to go.
Well, I hope this is the the same program.
That's okay.
Anyway, also want to thank John Leach, who has assisted me with the slides, and also Dave for his technical abilities. And I want to thank Rotary for everything that you guys do, both locally, nationally, internationally. And when I was invited to this, I thought about an experience I had back about 10 years ago. Involved. Our church became involved with a Haiti mission, and I had the opportunity to visit the island, and this was just an initial exploratory visit to the island and of La Gonave in Haiti. This is a very impoverished place, and I really didn't know what to expect. But we got to the island, and in the larger city of Anzagale, we got a truck, and we took this truck up a road, up the mountains, which was not really a road, but more of a riverbed, if you can imagine driving up. And it took us about 2 hours to go about 5 miles. And we came upon a clinic that had been built about 20 years before by a missionary named Bill Rice, who was a Presbyterian missionary and had been retired at the time and just felt the call to aid this island off the coast of Haiti. And so I toured this clinic that he built, and I came upon a plaque. And this plaque was the plaque from International Rotary that had a Rotary International who had donated all the solar power for this little clinic in the middle of nowhere that allowed for thousands to be treated on this island. So I thought it was very interesting, the importance of the work that Rotary does both locally and internationally. I'm just going to give a little background on the coronavirus at this time, just as an introduction. Back in 1965, a virus was isolated that was presented with common symptoms, common cold symptoms, cough, congestion. And scientists determined under electron microscopy that this virus had a certain structure, and they named it the coronavirus. There are about 4 strains that have been isolated of this virus, and it's been known to cause cold symptoms. It causes bronchiolitis in young children and flare-ups of asthma, even in children and adults. There was really not much attention to this until 2003. A novel coronavirus was discovered, which caused severe acute respiratory symptoms. And this virus was named the SARS-CoV virus. It was primarily just contained in Asia and other parts of the world, and the US. This probably came from civet cats to humans. Now, this particular virus really ramped up our preparedness because there were 774 deaths out of 8,098 cases. This was about a 10% death rate, which was very concerning, but it did finally come under control. There was a huge global response from the CDC and WHO, which really became our blueprint for further responses and to our response to the Wuhan virus. The MERS virus, this is the Middle Eastern Respiratory Syndrome, was discovered in 2012 and presented in the Middle East with, again, very severe respiratory illness, but it had a 40% fatality rate, and only 2 imported cases made it to the US. Now, this is our little bug that we've been dealing with, and you've probably all seen it with the spike proteins. And these little spike proteins, to some scientists, look like jewels on a crown. And so a corona is the Latin word for crown or wreath, and so it got its name. Now, this little booger is 2 millionths of a meter in size. That's about 1/100th the size of a common bacteria. So you do the math and see how many would be. I was trying to figure out how many grapefruit might go on this, and I couldn't do the math, so I figured. Moving on. December 2019, you all recall, the investigation of a cluster of pneumonia cases in Hubei province, which linked exposure of this virus to a seafood market in Wuhan City. By the 24th of January, Chinese scientists showed an 85% match with a SARS virus that's found in bats. So this was felt to be a recent emergence from an animal reservoir. And of course, the rest is history. Now, just a little bit about the Pfizer clinical trial. I'm not going to talk about clinical aspects of this disease because we've all heard about it, but focus on the vaccine trial and how we came to participate in it. Before I dive in, I want to discuss a little terminology. What is a clinical trial? These are experiments or observations done on humans designed to answer specific questions about biomedical or behavioral interventions. These protocols are used to provide data on efficacy and safety. Now, all of this is monitored and approved by an independent review board, or what's called an institutional review board, and gets plenty of oversight, both FDA and these institutional review boards. Now, as far as the nuts and bolts of a clinical trial, usually these start with preclinical phase. This preclinical phase is usually when we introduce animal models. Now, certain animal models mimic what a human response. So that's why many of these substances we use are tested first on animals to make sure that these are safe for potential human use. Then we go to phase one. Basically, healthy volunteers are asked to come into these trials and just to ensure initial safety before they are widespread. Just usually a few volunteers, it can be 40, 100, but before they get into large, widespread use. Phase two, this is a dose finding where you have multiple doses of a substance or a medication or product that can be and tested to see what seems to work best within certain safety parameters. Now, phase three are most of our trials. Phase three are actually when a drug company or a sponsor is trying to obtain an FDA approval. And so usually these are the trials that are most important to the drug companies in order to get them approved for market. In phase four are post-marketing trials, and there are many reasons why phase four trials are needed, particularly if there's a new indication. In other words, we find out that some product is helping in some other realm. Also, certain safety signals come up as we use these medications, and the FDA will ask for further studies from pharmaceutical companies to rule out or rule in safety issues. Now, as far as new indications, one of the examples that I think was real important was when Lipitor came out, and Lipitor, as you know, is a medication that lowers cholesterol. Well, it became pretty apparent that Lipitor and some of the other statins had protective effects beyond just what we thought was the cholesterol reduction. And so many studies were done on these statin drugs to show that they did, in fact, reduce cardiovascular disease over and beyond what their cholesterol lowering abilities were. And we believe this is probably reduction of inflammation in the body and in the vessels. That'd just be an example of what a phase four trial would be. Now, Piedmont Medical Group research, actually this is Piedmont Medical Group, PMG research, started in Winston-Salem. And this was begun by a physician named Tommy Littlejohn, who was a family physician in Weston in the 80s. And also along with Charles Hooser and Jennifer Byrne, who founded this company to do clinical trials. John actually retired from active practice. Tommy Littlejohn retired from active practice and then started just full-time work in clinical trial work. In the mid-90s, we established a satellite clinic in Hickory, and I was involved along with Carlos de la Garza and Dr. Bob Glenn, and we formed what's called PMG Research of Hickory. Since then, we had teamed up with Piedmont Rheumatology, which we currently, they have been with us from the start doing trials in rheumatology. We have a collaboration with Catawba Valley Medical Center. We're currently working with a woman's view on certain studies, and we've done some studies with neurology associates in the past. Dr. Scott Polster and Eric Clark are working directly with our site, either as primary investigators or sub-investigators on some of our trials. They're both involved with the vaccine trial. We have 6 employees, which include our site manager, Shay Perry, and she currently co-manages a site in Bristol, Virginia, and commutes back and forth. And she's currently involved in Bristol in the Novavax trial. Now, our outpatient trials include many sponsors, including AstraZeneca, Pfizer, Merck, GSK, Bristol Myers Squibb, Regeneron should be on that, and there are quite a few others that we have been involved with through the years. Our indications include, and these are just a few: diabetes, hypertension, osteoarthritis, weight loss, smoking cessation. Anybody getting an echo?
Yeah, a little bit.
I'm getting a little bit of an echo. I don't know why. But anyway.
I think I got it.
Okay, rheumatoid arthritis. And as far as vaccine trials, we've done pneumonia vaccines, flu, shingles, C. diff, and now the COVID-19. Now, in 2015, the whole PMG research network, which are about 12 sites, was sold to ICON. ICON is a large, it's a contract research organization. So what ICON becomes a middleman between the various sites and the sponsors. For example, Pfizer contracts with ICON, and then ICON contracts with the various sites to do trials. For example, ICON would do monitoring, a lot of monitoring of the trial for Pfizer. They help set up protocols. They help with a lot of safety monitoring. So they become an intermediary that Pfizer uses to do a lot of the nuts and bolts work of a trial. And they become experts at doing this. Let's see. Now, we now have 14 sites in 5 states in the US. And another bug in our issue with our setup is that ICON changed PMG's name to Accellacare. So we're actually now called Accellacare, which happened in 2020, along with a bunch of computer changes. And it's been very difficult from that angle because I lost all of my bookmarks, everything I had in my computer this year. So it's been quite a transition in terms just in a name change. It was not good timing. So currently, in terms of trials going on for vaccines, there were probably over 170 trials started worldwide. The one we are doing, of course, is a messenger RNA virus, but also Moderna, and 12% of all the trials are mRNA. There's also what are called protein subunit vaccines, where companies develop a protein similar to the spike protein, hoping to induce immunity. Vector vaccine, this is where you actually insert some information, RNA, DNA within actual a virus. I mean, actually within another virus, which is inactivated. For example, adenovirus, which is a cold virus. And you put this information in the virus, and it's not infectious, but it does. The body does recognize it and can make antibodies. And the others include DNA vaccines, and activated viral vaccines, activated virus-like particles, and replicating viral vaccines, which they do replicate, they carry material, but they don't cause infection. I say this is not AstraZeneca. AstraZeneca actually is one of these vector vaccines up here. This is a mistake. And anybody, if you want to read about this, Nature Journal, 13th October 2020, everything you want to know about COVID. Now, in terms of vaccine development, this graph is just a very schematic of those first three that I discussed. Down here is actually the mRNA, the messenger RNA, which obtained the genetic material. And and they make a vaccine out of this messenger RNA that codes for the spike protein. You inject this, and the body actually produces the spike protein. It looks just like the spike protein on the virus. And then the immune system produces the antibodies. So it's a pretty neat system of fooling the body into making actual virus spike protein. Another way is with the protein-based, actually you synthesize a protein that looks like the spike protein. This is incorporated into a vaccine. And again, the immune system produces antibodies. And I believe Novavax is working on this type of particle. Now, AstraZeneca, this is actually using the adenoviral vector. A gene is purified, and it's incorporated into the adenovirus. And the adenovirus gets into our cells, and the body produces spike protein, and again, the immune system produces antibodies. So these are very unique ways of trying to accomplish that process. Now, as far as Pfizer and BioNTech collaboration, BioNTech was a relatively small company in Germany that it was primarily involved in cancer research using the messenger RNA platform for cancer. And they were obviously, and then probably in February, they had actually worked out the genetic sequence that they needed to make for the messenger RNA, and were actually working on the vaccine. Became aware to them that for them to actually market, distribute, or pay for this development, they needed a big partner. And they collaborated with Pfizer. What is interesting, and there's a 60-minute segment about this recently, and also Time Magazine, the recent current, discusses the fact that the lead scientists for BioNTech, which actually owned the company, were very familiar with the lead scientist, vaccine development scientist at Pfizer. So they already had some connection, which was very helpful in moving this along. Now, this is kind of a busy slide, but it's just kind of the approach that BioNTech took with they derived the genetic information already which had been developed and developed a vaccine. And they came up with 20 vaccine candidates. This is probably in May, June. There we go. And then they did their preclinical testing with volunteers. Well, actually, preclinical testing, probably animals, and then clinical testing with phase 1 came over here. They had, by the time for their phase 1, they had 4 potential vaccine candidates. And so by the time and this was in July, and then the clinical testing phase 1 and 2 started early August, and then by mid-August, this is where we started in phase 2-3. Now we ended up with 263 candidates out of the 43,000 some that Pfizer had. But so they actually increased from 30,000 to 43,000 in order to get the numbers that they needed. Now, in terms of our collaboration with Pfizer, how we got involved was actually since we were then owned by Icon, Icon had picked 7 of our sites to do this study. And there was a lot of back and forth in terms of budget. There is on every study, negotiations for establishing what Pfizer was going to pay Icon for this. And finally, everything was set in July. Our trial began mid-August, phase 3. We didn't have really any trouble in terms of our recruitment. This was extremely busy time for us. The recruitment and the way it went out was really word of mouth, social media. We have a large database that we use, and so it was not very difficult to get it going. And here's the the results of that: 263 out of 43,548. Now, the actual how we did this study. Well, first of all, before we could do anything in Hickory, we had to be approved by a safety committee, which, interestingly enough, Paul Caparosi and Lee Young were on. And before they had to give us the green light on our standard operating procedure, which is about a 15-page document, to establish the safety of our site, the blinding, unblinding processes, and all those things that go into a vaccine trial. And my thanks to Paul and Lee for doing that. There's a consent process. Now, a consent form, which is, again, about 15 pages long, I think. Is that right, John? Was sent either email or people would read it when they come into the clinic before we could get to the next step. Now, once they signed their consent, we could take them back, and we had 2 coordinators, and one primary coordinator and one helper, who would then take the patient back and discuss all the various things we needed to discuss to obtain eligibility. And once they had established that and obtained all the data they needed and all the information, then I would go back to ensure eligibility, review the source document they were using to obtain the information, do a brief clinical exam at that time. Then blood was drawn, swabs were taken to test for COVID, not immediate swabs. These were sent off to another laboratory. So we really didn't know whether they had COVID or not when they came in. And then once all that was done, we had two vaccinators, one to give the vaccine, another was really just there for safety to ensure that what was being done was being done properly. And then the subject would go out in the waiting room and sit for 30 minutes to make sure that they did not have an adverse response or an allergic reaction or anything of that nature. And we really ended up, didn't have any real issues at those visits. Now, we did give our last shot. The last vaccination was mid-November. So we went from started in August, the last shot was mid-November. As far as what our experience, we had very few side effects. And I think most of you have probably read about this on people have talked about it, but there was some degree of fatigue that we noticed, a headache, some occasional fever, but not much, and some muscle aches. We had a few people with GI upset, and most of these symptoms lasted only 1 or 2 days. Of interest, we did these, as I mentioned, we did these swabs when people came in. We found out later, a few weeks later, that we did in fact have a few COVID carriers that did come into the office, but nobody was ever developed COVID from these visits. We had no unexpected. You've heard of things like severe allergic reactions and things like that, but we had no real unexpected issues on follow-up of the vaccine. Now, in terms of what Pfizer noted in the results of their trial, it was pretty amazing in that out of those, when they checked people 7 days after their second vaccination, there were only 8 people that developed COVID in the active group at 162 placebo. And that's where they came up with this 95% efficacy rate. So it's obviously somewhere, depending on your age, somewhere between 90 to 100%. They really did not find a lot of difference between gender as far as these results. The efficacy seemed to hold up for older people, seemed to hold up across the board pretty well. What's also interesting, after the first vaccination, 9 people in the placebo group developed COVID, and only 1 in the active group. And this was 7 days after the first vaccination. So there is even some, probably 50% coverage after the first vaccination. Now, one thing that's happened is, when you're doing a vaccine trial, you hope that everybody is blinded to the vaccine or the extent of the trial. Obviously, that was not going to work. Once in November, that when Pfizer received their emergency use authorization, then they felt it was their duty, and we all agreed that they should open up the study to the placebo group. And the way they've done this is to open it up to phase it based very similar to the phases of the CDC. What they have asked us to do is open it up according to our local public health. And so we have now unblinded our first responders, nurses, or actually all of our healthcare workers, and and also the 75 age group have come in for their vaccinations. And what I understand from Jeff is that we may be seeing the 65 and over group be opened up as well. And I'm going to be open for questions, but I thought I'd just answer a couple that may come up. Some people have asked, will this be an annual injection? I think the jury is out on that, but there's no evidence that suggests it will have to be, since at this point, although these new variants that are coming up still seem to be responsive to the vaccine. So, and it doesn't seem to mutate as quickly as, for example, flu vaccine, which you have to get a new vaccine every year. But I think it's possible that this will be an annual injection, or maybe biannual, or every 2 years. We have found, as far as antibody levels, we've had very good increase in antibody levels, which is called humoral immunity, but we've also had good tissue cell immunity as well. Probably as good or better immunity than the disease itself. And of course, distribution issues that you've all realized that this has had a very difficult time getting out. It's not an easy thing to manufacture, so but I think the distribution issues will be solved. It sounds like there's a lot of effort being put toward this. So I think in the next few weeks, this will turn around. So I'm just going to open the floor to anybody that may have questions, concerns.
With the trial.
I'm sure it's clear as mud.
So John,
Yeah.
John, this is Lee.
Yes, Dr. Young.
So you called in the people that you all unblinded, and called in the people that got the placebo to give them exposure to the vaccine so that they would become immune also?
Yeah, all the folks that are included in that first phase of the guidelines, first phase of the public health guidelines.
As far as, did you have any people that got the vaccine that actually eventually have gotten COVID?
I don't know of any.
Good.
I don't know.
And the COVID too.
I don't think so.
I'll say that what John referred to earlier in terms of maybe opening the studies or the vaccine up. There was an article in the Washington Post today saying that approximately sometime this afternoon, Alex Azar, who's the Health and Human Services, is having a conference with governors and going to suggest that they open it up to everybody 65 and over, and they're going to release all the vaccines and not hold any back for a second dose, similar to what Vice President Biden has suggested.
Yeah.
John, this Clarence Pugh.
Hey, Clarence.
What have we learned through this experience and the warp speed with which this vaccine was developed that will help us go forward in similar needs, hopefully not this serious, but do you see some improvement in timetables from turnaround and clinical trials, et cetera, that will make vaccines more readily available in a shorter time period?
Yeah, I think that a lot of the procedures and way things have been done, they have found, now, for example, in order to get this trial done, we had to obtain data and then upload it the very same day, everything. And that data was then reviewed and monitored. And then by the next day, we had questions regarding that. Well, that's a 2 or 3 week process that they were able to do, probably because they hired people to work all night long, through the night to get things done by the next day. So I think there'll be some streamlining in the future. I think, and a lot of these platforms, for example, the messenger RNA platform itself is ideally suited to coming up with very quick. If you can get we can get the genomic information, boom, we can make a vaccine.
That they were COVID carriers. Did they continue with the study?
I'm sorry, I didn't hear that.
You may have heard me.
I can't.
John, I have a question. It's
Yeah.
Lynn. Once you've had both shots, are you pretty much protected, but can you pass it on? Can you be a carrier?
Good question. I don't think so. I mean, if you had COVID at the time of your second vaccination,
That's my question.
yeah, possibly you could. But I would say that that would be very unusual.
Okay.
That'd be the only scenario I think that would happen. Now,
Okay.
it's possible. Obviously, there's a percentage of 5% of people who got COVID regardless of the vaccination. So it's still there's a 5% chance, but 95% is pretty good.
Yes, it is. Thank you.
Mm-hmm.
John, this is Shuford Abernathy, and this may be the same question Lynn asked, but in just a little different way. That once you have the vaccines, can you pretty much go back to living a normal life?
Do what?
Once you've had the vaccines, can you go back to pretty much living a normal life?
Well, that's a good question. I think you still need to take precaution because there's so many. When you think already 10% of our population has had COVID over the past, and another 10% in the next 6 months, I think you still need to be careful. I still would assume that the person across the room may have COVID until we get really good herd immunity. Now, as far as the herd immunity, one thing says we need to have 70% people vaccinated, but I think we need 50% vaccinated, because by the time we get to the summer, 20% of our population will have had COVID. So we'll have that 70% of that immunity.
Dr. Earl, this is Michael Blackburn.
Jeff, if
You may have answered this, but if you've had COVID, do you need to have the vaccine?
That I think that's going to be a personal preference. If it were me, I'd probably just go ahead and get it.
Mm-hmm.
But yes, because I would wait. I mean, if you had COVID, I would probably wait a month or 6 weeks before you got your vaccine.
Yeah.
Since it's available, it's free. Why not?
Yeah. Thank you.
Yeah.
Dr. Earl, Alan Barnhart here. A little bit variation of the question. If you have had COVID, but not the vaccine, are you a carrier for other people? Could other people catch it from you?
No.
If you've had COVID, but haven't had the vaccine?
Right, and you've recovered, are you still a carrier?
Not necessarily. It's possible. I mean, I think there are people who may have that carrier state, but if that, it's, again, that's very, very small.
Thanks.
Now we have, it's, go ahead. Who's next?
John?
I was going to say,
Yeah.
if you look at your chat feature, there's a question in the chat feature.
Is there?
Yeah.
Oh, yeah.
He's paying for it.
Question is, who's paying for the vaccine?
Oh, okay. Oh, that's a good question, because the actual production and manufacture of the Pfizer vaccine was all Pfizer money. There was the program called BARDA, where the government gave money to pharmaceutical companies to assist them with production. Pfizer did not take any of that money. Now, where there has been, Pfizer did accept money for the distribution. I mean, all the companies of the government provided money for distribution of the virus, but the initial manufacture, all the research and development, all that was paid by Pfizer. None of it from the U.S. government. We got one. Kudos to Chief Lucas for his letter to the HDR. Now, I didn't have a chance to read that. So, somebody want to comment on that?
Probably not.
Okay.
Yes, I did that. He had a wonderful letter commenting on the incident at the Capitol. If I saw him, I was going to say thanks for that letter, and since I won't see him in person, I took this chance.
What now? Just tell us which day was that?
Tuesday, I think. I mean
Thursday. Okay.
Dr. Earl, one more question. Do you see the reluctance of healthcare workers in getting the vaccine as being overcome over time? Are they just being careful?
Would you say that again? I'm sorry, Mike.
I'm understanding that healthcare workers are somewhat reluctant, or at least were disappointed in their involvement in the vaccine. Do you think that's going to go away as they get more confidence in it, or has anybody got a strategy for how to make that better?
The healthcare workers were not confident.
Yeah, there's a significant number of healthcare workers that have not opted for the vaccine.
Those that,
it's being offered and they're there and they're probably expected to do it, but they don't do it.
Yeah. That's going to take some time. There are a lot of questions that people have about the vaccine that I personally think have been answered, but I know a lot of people's minds, there's still questions.
They think they know too much.
Exactly.
No.
John, one more question. How about the other people that were running their studies? Did they have any significant effects from the vaccine? Say if a person was a COVID carrier and inadvertently got a dose of vaccine, being a carrier, did any of them have a hyperreaction to the vaccine?
I haven't heard anything like that.
Okay, good.
I wanted to make to Mike's question, Michael's question. There are things you're going to read about how these genetic substances will be incorporated into our cells, and therefore it's going to change us genetically. I mean, I've read many articles on people concerned about that, but this messenger RNA does not enter the nucleus of the cell. This messenger RNA, it goes in, it does its thing, and then the cell gets rid of it. So there's no genetic encoding of anything within our own nucleus, so of our cells. So,
John, just one quick comment that I would.
Go ahead, Paul.
Messenger RNA technology is such that this is probably going to revolutionize the practice of medicine, particularly as it revolves around cancer treatments, because if they can get genetic codes for a particular form of cancer, they may well be able to allow the messenger RNA to transmit that to our immune system. So I think it's very encouraging technology. It's obviously new to us, but it's going to go strong. Thanks.
Dr. Earl, this is Lou Wetmore. Thank you so much for that program. That was very enlightening, and I think that it answers a lot of questions that we all have about this terrible pandemic and the disease, and the hope that we will get through it at some point. David, if it's possible, we've used our hour up, so we really need to close out. But if you want to leave this open for other questions for Dr. Earl, and he's willing to stick around a while, I think that would be just fine. If you can do that. Now, in the meantime, as was mentioned earlier, we will not have a meeting next Tuesday. We will be meeting with the Thursday Club on the 21st, and they will be hosting, and sounds like they have a really nice program for us that day. So if y'all will please join me in conclusion with the four-way test, I would appreciate it. Of the things we think
Say or do. Is it the truth? Goodwill and better friendship. Thanks, John.
Dr. Earl, thanks.
Thanks, Dr. Earl.
Thank you, John.
Glad to do it.
Thanks, John. Thanks
Very
a
well.
lot.
Thank you very much.
Thanks, John.
Bet.